We're out of time and we're going to go ahead and move into the regulator panel discussion. So I'll go ahead and introduce all the panel members up front, and then we'll jump into a short presentation from them all before we get into the discussion questions. We'll have Dr. Borkowski present first, and then we'll go alphabetically from there. So again, just a reminder to enter any questions or comments that you have in the Q&A. So with that, I will have Dr. Gary Borkowski speak. He's the Global Director for AAALAC International and has been in this role since November 2018. He's responsible for overseeing the accreditation program for AAALAC International. We also have Dr. Suzanne Fitzpatrick. She's the Senior Advisor for Toxicology at FDA's Center for Food Safety and Applied Nutrition. And Susie is a board certified toxicologist. Next, we have Dr. Nicolette Petervary. She's the Director of the Division of Policy and Education in the Office of Laboratory Animal Welfare at the National Institutes of Health. And she was formerly the National Animal Welfare Specialist for Animal Welfare Operations with USDA APHIS Animal Care. And then last but not least, we have Dr. Gwen Maginnis. She's the Non-Human Primate Specialist with USDA's APHIS Animal Care Center for Animal Welfare, which she joined in 2015. And she assists stakeholders and inspectors on primate care and welfare. And so with that, I'll hand it over to Gary to start us off.
Thank you, Jesse. Can you hear me okay?
Yep.
Okay, great. Thank you, everyone, for having AAALAC be part of your meeting. As Jesse said, I'm going to give a short presentation, and then the Q&A will be handled after the subsequent speakers talk. So I'm going to start off with a slide here that may be a reminder, it may be new information. So last year, about a year ago, actually, AAALAC convened a strategic plan, a new strategic plan, and you may have seen some action as a result of that. One of those actions was to develop a new vision for our organization, which is posted at the top of this slide, and I'll read it. A world where excellence in animal welfare and science converge. We also adopted a new mission statement, which reads, AAALAC International improves the welfare of animals in science and education through the accreditation of organizations meeting high standards of humane and responsible care. So today I'm going to talk a little bit about where AAALAC is in regards to the 3 R's. And I'm going to start with a bit of a history lesson. I'm sure this is not new information for most people. You can see on the left here, the seminal book, The Principles of Humane Experimental Technique. I know Steve mentioned this a bit. You can see in the lower right corner of that book, it was published in 1959. And then on the right, you can see the 2 gentlemen responsible, Birch and Russell, which we hear a lot about. So where has AAALAC evolved since that 1959 date? Well, one of the notable things that we do is we offer a Global 3 R's Award program. And here you can see some of the information about that program. Importantly, you can see that if you receive one of these awards, you can receive up to $10,000. And we have 4 awards each year, one for North America, one for the Pacific Rim, one for Europe, and one for the rest of the world. So, you know, calculating that $40,000 a year could be giving out. To me, that seems like AAALAC has committed to and is supporting the 3 R's as a concept. So if you're interested in looking more into this, you can see there's an online application. Just go to our website, aalac.org. Or if you prefer to download an application, you can do that from our website. And importantly, the deadline is June 1st, so time is ticking. And again, I would encourage you to look into this. And if you don't think you have a peer-reviewed paper that would qualify, please share this information with others, and maybe they'll give you a cut of the prize money. All right, so this is a bit of a reminder for the audience of what information is used when a program is visited for accreditation. So I'll start with some very basic information, our rules of accreditation. Always a good reading. If you're having trouble sleeping, you can go to our website, click on the link, Rules of Accreditation. And one of the important things in there it talks about is that we expect programs that are accredited to apply with statutes and government regulations in the geographic location that they're in. As I think most of you know, we also apply primary standards. Currently, that's the 2011 guide, the 2020 Ag Guide, and ETS 123. In addition, we have a variety of documents we refer to as reference resources, and I'll talk a bit more about those and give you an example of where the 3 R's have been incorporated into that. We have many frequently asked questions, abbreviated FAQs, and you can again review those. We're always updating, revising, adding new FAQs as relevant to the evolving animal research space. And then finally, we have position statements. And again, I'll link to those in a minute here. So starting off with the 3 primary standards, for the sake of time, I'm going to just give you a couple examples from the Guide for the Care and Use of Laboratory Animals, which is actually referenced in our rules of accreditation. And you can see the citations here on the lower part of this slide. Secondly, just to pull out a little bit more information from that primary standard, and these are either quotes or phrases from the guide. So the first quote is, "The 3 R's are recommended as a practical strategy for decision-making for the use of animals." And then it goes on to talk about specific examples. And so I'm not going to click on these links, but you can see the first sub-bullet there, humane endpoints. We do have an FAQ on that topic. And this is, again, how AAALAC has chosen to support and advance the 3 R's. One of the phrases within this section of the guide talks about if there is pain or distress, veterinary consultation should occur. And so the second link will actually link you to our position statement on the attending veterinarian and adequate veterinary care. Another statement from the guide is, over the years, the 3 R's have become an internationally accepted approach for researchers to apply when deciding to use animals in research and in designing humane animal research studies. So again, this is a quote that is part of a standard that we apply to our accredited programs. And then finally, a phrase, it's a section of a sentence, "The AWIC is obliged to weigh the objective of the study against animal welfare concerns." And so that prompted, in part, a FAQ that we now have posted on harm benefit analysis. All right, so now I'm going to pull out a little bit from a FAQ that we posted, revised or developed and posted a couple years ago. And the topic is scientific reproducibility. And so if you look at the second blue box here, just to explain, when it says in either scenario, the FAQ refers to whether you have an internal review for scientific merit, if you will, or external. But then it says the AWIC or OB, which stands for oversight body, should confirm that the protocol contains pertinent study design elements and ensure adherence to the principles of the 3 R's. And then these are some of the other areas that are focused on in this scientific reproducibility FAQ. So you can see on the lower left, justification for the selection of species and animal numbers. So that would be relevant to the 3 R's. On the upper right, less invasive procedures. So that could refer to refinements. The next box, consideration of alternatives or lower use species, so replacement. And then the other phrases here also are relevant to advancing humane animal care and welfare. All right. So I mentioned earlier that one of the sources of information that we use for our accreditation program are reference resources. Again, I would encourage you to get familiar with these on our website. These may be used in conversation during a site visit by the team. They could also be used during the council deliberations as the accreditation status and a specific finding is discussed. So you can see at the bottom here, just to help you understand the genesis and basis for reference resources, the statement says, since 1975, AAALAC International has also referred to other specialty publications for supplemental information about procedures or techniques related to the care and use of laboratory animals. These specialty publications are designated as reference resources. So on this slide, you can see on the right, one of the reference resources we have is the Institutional Animal Care and Use Committee Guidebook. And then within that guidebook, I took a snip from the table of contents, and under section C, C2, C2A, there's a section on alternatives, replacement, reduction, and refine. And so now I'm going to kind of back up and look at the big picture here. To me, this is a good example and a reminder that to show you how all these things interconnect. So in 2018, I had the opportunity to attend the OLAW eye care session. And during that session, the topic that me and my team focused on was the 3 R's. And this was one of the slides we developed from that presentation at the eye care conference. And so to me, you can see here good examples of how scientists are working to help improve the welfare of animals that are still necessary in scientific research. And so the very left side are the data points. The second column, guinea pig maximization test. You can see a great reduction from that test to the local lymph node assay, cutting from a month to about a week. The number of animals has been decreased from 30 to maybe a dozen up to 20. And then from a refinement perspective, dermatitis is not induced and Freund's adjuvant is not required. And so with that, if the audio works here, I'm going to let one of the authors of the book leave us with some closing comments and encouragement.
You've got to accentuate the positive, eliminate the negative, latch on to the alternative, always replace, reduce, refine. You've got.
And with that, Jesse, I'll turn it back to you. Thank you so much.
Great. Thanks for the presentation, Gary. And now we'll pass it over to Dr. Fitzpatrick, and she'll give an update or a short presentation from the FDA.
I sent you my slides, so I don't have them. But
Okay.
while you're looking for them, I will say that former speaker had a misimpression of the recent legislation that came out on animal testing as regards FDA, which has been a common misimpression. FDA has never had any requirements for animal testing. There were some guidance, but guidance is just our best thinking. We have always accepted alternatives as long as that we have confidence in the data. So the legislation that came out really didn't change anything as far as microphysiological systems. We began that program, began that program seriously with DARPA in 2011, and we put a significant amount of resource into it ever since. We haven't seen a lot of data coming into applications, but that's really, we don't have any choice as to, we can recommend, but can't require what people put in. So I hope that helps you understand a little bit more what's going on at FDA. I will go on to the next slide. Can I move them myself, or can you turn this?
You can just tell me, I think.
The next slide, yeah.
Okay.
So I'm just going to talk today about, I'm in the foods program, and I'm in charge of the new approach methods foods program for Sysem. But I can say that FDA as a whole has an alternative methods work committee, and we have senior members from each of our 6 centers, product centers, plus the field, the field force, and the Office of the Commissioner, and the Office of Terrorism, and One Health. So we all work together and to talk together about looking at different ways to do alternatives. But we recognize that each center has different products. Our products, we lack, for most of the products that's in the FDA foods program, we lack pre-approval authority. And what does that mean? That means that if we believe one of these products that we regulate, other than direct food additives and some packaging, is causing problems in the food supply, we have to develop the research in which to demonstrate the problems according to label use that a product is causing, according to label use, and then have the authority to remove it from the market. So that's a pretty big burden. The burden is not on the sponsor of most of these products to show safety. The burden is on the FDA for most of these food products to show harm. And that means, generally speaking, we do research ourselves. And that means if we see harmful substances in the food supply, and we do see them, we cannot tell our consumers that we're going to go back and do an animal study, and it'll be a couple of years before we can tell you whether it's really harmful at the concentration we see. So developing these new approach methods is very essential to helping us assure there's a safe food supply for everyone. So, and with that, we've developed our own new approach methods program. And I think the next slide talks a little bit about it. Okay, so in 2017, we put out the FDA Predictive Toxicology Roadmap. And this was really a way, came out of the Office of the Commissioner. It was really a way of showing step by step how we were going to implement new, more predictive methods. And I chaired this committee, and now we have put these principles into action in the FDA Foods Program. So the framework for this is to show that it was and really in some sense we were in competition with EPA since they had put out a roadmap. We were going to invigorate and strengthen FDA's long commitment to promoting, developing, and use of new technologies that better predict human, animal, or environmental principles. And we in the FDA Foods Program, our Suzanne, have incorporated these principles into our new approach program for food safety assessment. At the same time, two other important roadmaps came out. The Tox 21 roadmap, which is a partnership between FDA, Suzanne, EPA, NCATS, and NIH to work on new predictive methods. And then the ICFAM roadmap also came out, and all three of them not only stressed 3 R's, but we stressed what we call the 3 C's: commitment, communication, and I'm blanking on the third one. Commitment and communication and another C. So what it really means, collaboration. So we're going to collaborate together. We've committed to go forward with this, and we're committed to finding a result. And so this is that those 3 C's besides the 3 R's are woven into our roadmap, into the Tox 21 roadmap, into the ICFAM roadmap, and right now we've also updated the ICFAM guidance, and that's out for the guidance on qualification, and validation of new approach methods, and that's out for approval by the 17 ICFAM agencies. So, and FDA also chaired that. So next slide. So these are the roadmap steps, and I can go through each one. There's 6 steps in this roadmap, and we use those steps to design our program. And in actuality, we are asked by Congress quite a bit of what are we doing about the roadmap, and all the various different steps in it. What are we doing about this? What are we doing about that? So people continually ask us, including our Congress, especially what are we doing to train our regulators, and what are we doing to work with our stakeholders? So next slide. So the first one was to develop a senior level working group. So we developed under the Office of the Center Director at Suzanne a NAMS working group to leverage our resources, and we have on the working group both regulators and researchers, both senior and junior scientists are part of this. We have a liaisons with the NCTR and with the field. So it's kind of going on its own, I guess. So maybe we can back up a bit.
Yeah, sorry, it has a mind of its own.
Okay, yeah. So next slide then. This is our charter. And this is a charter that we have actual deadlines for. We're going to develop criteria for having confidence in new approach methods. We are identifying what is being used in the food industry right now in academic and other government agencies. We're going to develop a plan for the applicability of these for evaluation that includes qualification criteria that we will publish, feasibility, how much they cost, where that method is in its development stage. We're going to we'll publish a qualification guidance, probably like a notice and comment, and we will conduct workshops, including what we call case studies, where we have a case study and we all get to try to apply the criteria and see how really functional they are. So next slide. Next was training of research regulators. We are trying to make sure that, first my boss said, don't be offense, but you're not going to be here forever. We have to make sure that other people know what's going on. So we're really doing what I think I call training by doing. We include junior scientists in all of our planning and implementation of NAMs, promising people, we're giving them more opportunities than maybe they would have gotten. If someone asked me for a talk and I think someone else can do it, I'll help them give it. We're working with the UK and we have 2 of our younger scientists taking the lead with support from our senior scientists. So we're really trying to get them involved and networking in this whole area. It helps their careers. It makes them want to stay at FDA and CivStan, and it helps us too with new and fresh ideas. So next slide. And this is we're encouraged people to partner with us. There was a partnership slide saying partnerships with our stakeholders are very important, and there's different ways to partner with FDA. You can have public-private partnerships where FDA is invited to participate. There is a convener that's a nonprofit, that's a convener that convenes a group of scientists across all the sectors that are interested in food to come together and talk about science issues. We do memorandums of understanding between federal agencies to share information and resources, and we are being encouraged by the OSTP and the White House to tackle all of these problems on a cross-agency basis. We do research collaboration agreements, which are we collaborate jointly on a research project, and I don't have listed here, but we do CRADAs, or cooperative research agreements, where technology is moved into our laboratory, and those CRADAs are what we use to move organs on a chip into a lot of our laboratories so that we can look at that technology, be familiar with it, decide what criteria we want to apply to it before we see it in an application. So almost all of our products, actually, except for devices, have organs on a chip, or MPS, and or organoids in their laboratories, working with their regulatory scientists to look at criteria. Next slide. We also have an international liaison group on risk assessment of chemicals in food. We partner with EFSA on it. And additionally, we have a roadmap for new approach methods for food safety. We have as partners with us not only the EFSA countries, but we have Canada, Japan, Korea, Singapore. We also have OECD and JRC on it. And next slide has, I think, our goals. So the goals, okay, food is a mixture. So we're looking at how do we look at mixtures using new approach methods. We're trying to get some new guideline type studies to have harmonized reporting and context of use of qualification approach. So context for use means you start up front and say, we're going to use this particular method or new tool for a certain use. Okay, so that replaces what in the past was sort of a one-size-fits-all validation. If depending on the context of use really dictates how much qualification criteria you need. If you have a screen, you don't need as much confidence in that method than a pivotal study coming through that might be replacing a whole animal study. So the context of use really helps define the qualification criteria that are applied to that particular thing. And that's part of a real revolution in risk assessment, which includes looking at mechanistic risk assessment, mechanistic validation, including AOPs and IATAs, seeing if those tools, by adding that mechanistic criteria, in other words, some potency, can really replace or supplement animal testing for concurrence validation of non-animal methods. So next slide, please. And we're committed to communicate with anyone that wants to talk to it. We're encouraging sponsors to come in and submit validated methods. We're encouraging people to come in and talk about methods. This is across the FDA, not just in the foods. We're encouraging discussions with stakeholders. FDA as a whole each program has been encouraged to either start or continue or reinforce these dialogues with their stakeholders. And we have this, and one of the ways we've done it, and I mentioned this before, was the development of a public-private partnership, which where we and actually CAD is on this public-private partnership with us, and we talk about science. CEDAR has probably 20 public-private partnerships. CDRH has what's called a collaboration communities, which are smaller public-private partnerships discussing things. But these are all of these ways are tools to allow FDA, any FDA, part of FDA, to have dialogues with our stakeholders. MOUs, RCAs, public-private partners, everything allow us to have dialogues with our stakeholders, industry, academics, NGOs, consumers, in order to put better products on the market. And for us, the goal of a safer food supply is everybody's goal, and so everyone giving and contribute. Everyone's ideas and contributions are important to us. So next slide. I think we also want to make sure our research actually identifies needs. So we want to identify what we're working on is identifying our gaps first, where we need new tools to look at regulatory questions, and then design the tools to answer those questions, rather than designing a bunch of new approach methods and then trying to fit them into a regulatory program. So next slide. And that's what we've done here. We have emphasized that the regulators must be included upfront in any new method development, new approach methods. They need to identify the gaps that we need research on, what tools are needed, and then they need to work with the academic developers as they develop those new tools. This is the model that we used between FDA and DARPA and CATS to develop the first microphysiological systems, the 2 grants we gave to Harvard and MIT. We met with them, and then several NCAS grants after that. We met with them monthly to make sure that the regulatory needs or gaps or ideas were incorporated in those tools, and then we followed it with CRADAs, bringing some of those tools into our lab. We also have another partnership with NCAS right now to develop a neuromuscular junction on a chip for botulism toxin testing. So we have talked to them. They are funding a research in an academic and an industry lab. FDA will be working with them hand in hand as they develop those chips, and then we are going to qualify or validate it against the Mountsley Valley test. So it's really a partnership between regulators and researchers at FDA to develop these new approach methods that we need to do our jobs better. And next slide, please. And I think this is probably the last one that when we wrote the FDA predictive tox roadmap, we really wanted to put in some accountability. A lot of things are written in government with no a senior level accountability put in there. And we never see them again half the time. So we put in there that for the predictive tox roadmap, they were accountable to the Office of the Commissioner every year to somehow indicate what progress we're making in alternatives. And we also have that accountability in the foods program. Not only do we have strict deadlines for each of those points that I said we had to do, but we are accountable to the Office of the Center Director and as our program. And I'm sure everybody knows that the foods program at FDA is under reorganization to have a deputy commissioner for foods, and we will be accountable to that deputy commissioner then for what we're doing in the foods program too. I think that was my last slide. But thank you very much. I felt a little bit like I should say duck out of water, a mouse out of water in the meeting because I work basically on non-animal methods and not animal methods, but it's always been it's interesting to hear a lot of what's going on. So thank you.
Great, thanks, Susie.
And I will say that CAT is a very good partner of FDA and especially Suzanne. So
Awesome. We look forward to seeing you on the discussion part. And now I'll hand it over to Nicolette from OLAW.
Hi, everyone, and thank you. Today, I'd like to present some OLAW perspectives on implementation of the 3 Rs, but first, I would just like to say that this conference is near and dear to my heart, precisely because we have so many different perspectives and we get information from people with different areas of expertise. And I really see this as a way for all of us to exchange ideas collaboratively and move forward. And I've learned an awful lot the last couple of days. But for now, I'm going to give you the OLAW perspective on the 3 Rs. And I actually did a keyword search in the PHS policy, and I looked up the term 3 Rs, refinement, replacement, and reduction, and surprise, none of those terms were found. But I would still argue that the meaning and the intent of our policies and guidance regarding the 3 Rs is there. And it's built into OLAW's foundations. And examples can be found throughout the policy and our guidance. And this is especially clear when you look at the US government principles. As a review and a little bit of history, they were developed in 1985 by the Interagency Research Animal Committee and subsequently adopted by US government agencies that develop requirements, as well as agencies that sponsor procedures with vertebrate animals. The principles were incorporated into the PHS policy in 1986, and they continue to provide an essential framework, and I would argue a relevant framework to this very day. So I'm a bit of a graph junkie, and so I put this together to prove to myself that all 3 Rs are covered by the US government principles. And you can see that easily here in terms of refinement. We have representation in US government principle 4, to avoid and minimize discomfort, distress, and pain. And the observation that pain and distress in humans may also, procedures that cause pain and distress in humans may also do so for animals, and that should be considered when it's not certain. We see that there are requirements for appropriate sedation, analgesia, and anesthesia for pain and distress in Principle 5. In Principle 6, they discuss euthanasia for severe and chronic pain or distress. And in 7 and 8, we talk about species-appropriate living conditions and veterinary care, as well as trained personnel. And for reduction, we see appropriate use to look at the benefit, species, quality, and the minimum number to obtain valid results in Principles 2 and 3. Minimization of pain consistent with scientific practices, and replacement. Actually, the government principles specifically discuss this, where they talk about use of other models, such as mathematical models, computer simulations, and in vitro systems. So I think this document was really ahead of its time in a lot of ways, and it is still a good guidance document and a good path forward even to this day. And even though the PHS policy doesn't use 3 Rs terminology, it espouses the same concepts as in the US government principles 4 through 8, as evidenced in part 4.1c. And you can see that they discuss minimization of discomfort, pain, and distress, use of analgesics and anesthetics, as we described before, euthanasia for severe or chronic pain and distress, and species-appropriate living conditions, training, veterinary care. All of those good things are also described in the PHS policy. The guide for the care and use of laboratory animals does use 3 Rs terminology on page 4 and 5, and specifically it talks about a very important aspect of welfare, which is discouraging animal reuse as a reduction strategy. And I think that's really important. And throughout the guide, there's discussions of other important 3 Rs concepts, such as species-appropriate housing, social grouping, physiological and behavioral health, minimization of pain and distress. And it emphasizes positive experiences and maximizing those, as well as minimizing negative experiences, which I think is really important to the modern science and application of welfare. It specifically discusses species typical behaviors, physiological well-being, exercise, manipulative activities, and cognitive challenges, which emphasize the importance of incorporating those positive experiences. OLAW provides guidance to IACUCs and other oversight bodies on the importance of the 3 Rs in its frequently asked questions, and you can see the URL on the slide. FAQ D6 refers IACUCs to the resources I've just described, as well as the institution's assurance and animal welfare regulations, as if they apply, as protocol review criteria. And D7 goes even further by specifically addressing the 3 Rs as important considerations in protocol review. In a word, should the IACUC consider the 3 Rs when reviewing protocols? Yes, they should. And it also refers to the US government principles as the basis for this guidance. FAQ D12 specifically calls on IACUCs or other oversight bodies to evaluate the relevance of a proposed procedure to human or animal health, the advancement of knowledge, or the good of society, as stated in US Government Principle 2. I keep going back to the principles because they encompass a lot of the key foundational concepts to animal welfare. In addition, PHS policy review should refer to sound research design, rationale for involving animals, and scientifically valuable research. And the FAQ states this outright. From OLAW's perspective, a study that could not meet these basic criteria would be inherently unnecessary and wasteful of the animals, therefore not justifiable. So I often get a lot of feedback that the IACUC is not tasked with evaluating experimental design. And to an extent that is true, they are not peer reviewers. That is different. And certainly they shouldn't be looking at the experimental design through the lens of a peer reviewer. What they should be doing is looking at experimental design through the lens of animal welfare. And in terms of doing a harm benefit analysis, looking at that is extremely valuable both for the welfare and for the science. You can find, once again, these FAQs on the URL at the bottom of the screen. So OLAW has other FAQs that provide information on social housing, positive reinforcement training, and outcome-based performance standards for housing and enrichment. OLAW has repeatedly reiterated that social housing for primates and other species should be the default and should be implemented unless there is compelling scientific, veterinary, or animal welfare justification for single housing. And as we've seen from Chloe Stevens' talk this morning, data supports this for other species. It applies to other species than non-human primates. And the more we learn about all of these animals, the more we see the benefit of complex environments, the benefit of social housing. An emphasis on performance standards means that enrichment should be outcome-based. And the goal for enrichment isn't providing an item or even multiple items on a checklist. The goal is to provide positive interactions that foster highly motivated, positive, species-typical behavior that give them good psychological health and positive affect. And hot off the press, NIH recently released a guide notice, and this guide notice is Notice OD 23057 that encourages the use of the ARRIVE Essential 10 for publication reporting on the results of both vertebrate animal and cephalopod research. Now, this is not a requirement, this is an encouragement, but I think this is an important step forward. And if you don't know what the ARRIVE Essential 10 is, I suspect most of the group that are attending do. It is a series of 10 items that need not only be considered, but reported. And I think of particular interest is item 9 out of the Essential 10 that discusses reporting the procedures used to develop the model. For instance, the induction of the pathology, the procedures used to measure the outcomes, pre and post experimental procedures, including animal handling, welfare monitoring, and euthanasia. And as a reminder, the Essential 10 are intrinsic to the experimental design, and therefore they must be considered during the development of the proposal to meaningfully benefit the science. They really won't work if they are put together as an afterthought. And just in case you're interested, OLAW will be holding an online seminar on the Essential 10 on June 15th, 2023 with Dr. Penny Reynolds, if you're interested in finding out more Those are free of charge to attend. And finally, OLAW has web-based resources, including web pages on primate enrichment and social housing. So I hope you'll go ahead and visit those. We also have a page with workshops and conferences that may be of interest that focus on national education and animal welfare. And with that, thank you, and I'm happy to take questions at the end.
Great. Thanks, Nicolette. And with that, we'll hand it over to our last speaker, Dr. Gwen Maginnis.
All right, thank you. Hi, I'm Gwen Maginnis. I'm the non-human primate field specialist with the USDA Center for Animal Welfare. And I'm going to cover the USDA perspective on the 3 Rs, or really kind of where the animal welfare regulations relate or support the 3 Rs. The Animal Welfare Act and regulations don't actually mention the 3 Rs or really directly refer to them or address them. However, there are many places throughout the regulations that refer to professionally acceptable standards or currently accepted professional standards. And USDA considers the 3 Rs to be part of those standards. There are also a few regulations that indirectly support or align with the 3 Rs, specifically in the sections on scientific protocols, staff training, and annual reports. So first I want to talk about replacement. And the content on all of my slides will come either directly from the Animal Welfare Act or the Animal Welfare Regulations, and I do have the section listed before the excerpt, or it'll come from our Animal Welfare Inspection Guide, which sometimes offers more details in terms of what our inspectors are looking for when they're determining compliance at facilities during an inspection. Both of these documents are publicly available and they can be accessed online at the URL at the bottom of my slide. Our requirement that researchers verify the need to use live animals in research is essentially a requirement to provide assurance that an alternative model would not meet the needs of the study. So that's one way that it encourages alternatives. Additionally, our regulations on training specifically mention providing guidance to personnel on alternatives to the use of animals in research. For reduction, there are regulations pertaining to both protocols and training intended to prevent unnecessary duplication of research with animals, which serves to reduce or at least avoid needlessly increasing the numbers of animals that are used. Researchers are also required to justify the numbers of animals on their study to ensure that the minimum numbers of animals are used. And our inspection guide provides examples of the types of rationale that our inspectors are typically expecting to see. For refinement, there's the requirement for researchers to consider alternatives to painful or distressing procedures. And in many cases, the consideration of alternatives tends to focus on techniques that are less invasive or the use of non-invasive procedures like imaging, which would be refinements. But there are times when the most relevant alternative may be a non-animal replacement or be something that results in reduction of the number of animals studied. As an example, the inspection guide mentions some of the alternatives to consider for toxicity studies, like the up-down procedure as a refinement for LD50 studies, and which would reduce the number of animals used on the study, or different non-animal models like tissue assays, which would be a replacement rather than technically a refinement. When procedures that cause more than momentary pain or distress cannot be avoided, or there are no alternatives that would meet the needs of the study, animal welfare regulations require that any discomfort, pain, or distress be minimized as much as possible. And when our inspectors are reviewing these types of protocols during a facility inspection, they do pay special attention to any measures that are used to alleviate pain or distress on the category D protocols. It's also worth noting that detailed instructions or criteria are necessary anytime the frequency that relief is provided, it is provided on an as needed basis rather than with a specific interval. And the reason why we require that is to make sure that the people who are providing the care to the animals are on the same page with the intent of providing that relief when it's needed. As it has been touched on in several of the other presentations during this meeting, it's not just the experimental procedures, but it's also the living conditions and the daily activities that have a significant impact on animal welfare. And so our regulations on what needs to go into a study protocol also include the requirement that living conditions are appropriate for the species and contribute to their health and welfare. And that they also align with the standards of care. And for non-human primates, that would include the provision of environment enhancement to promote psychological well-being. And last year, or no, it was earlier this year. It was going to be in November, but I believe it actually came out in February. We had an advanced notice of public rulemaking that included looking at our regulations to provide environmental enhancement to all covered species and not just non-human primates. So that's something that we are looking at doing, and that is very exciting. The annual reporting process also supports the 3 Rs, and it supports the 3 Rs through public transparency. Research facilities are required to provide assurance that each PI has considered those alternatives to painful procedures. They're also required to provide the numbers of animals that are used for research, categorize the level of pain those animals experienced, and provide a full explanation for any studies where medications to alleviate pain or distress are withheld. And then this information is made publicly available on the USDA website. And then I have one slightly more indirect topic that I wanted to touch on, and actually Nicolette touched on it as well. And that is the reduction of animal numbers at the expense of the individual. And by this, I mean things like animal reuse, particularly involving surgical protocols, which is a topic that has recently seen a lot more interest in light of the constraints on current non-human primate resources. Another example is the reluctance to socially house animals on a study because it will result in an increase of the animals associated with the study because of how the study is designed. And decreasing the overall animal numbers by increasing the detrimental impact to the animals that are involved is not consistent with the overarching intent of the 3 Rs to promote the humanest possible treatment of experimental animals. Relative to this, animal welfare regulations do not permit animals to be used in more than one major operative procedure for the sole purpose of reducing the number of animals used. Additional major operative procedures are only allowed if they are justified for scientific reasons, if the procedure is performed as part of veterinary care specifically for the health and welfare of the animal, or for any special circumstance that is specifically approved by the USDA on an individual basis. And on inspection, we're interested in how facilities are tracking their animal use to ensure that they're avoiding multiple major operative procedures. And if the facility has been granted a variance that allows them to do multiple procedures, we want to verify that the stipulations of that approval are being met. And that sums up the key areas where the animal welfare regulations sort of connect with the 3 Rs. And I guess we're ready for questions.
Great, thanks, Gwen. And thank you again to all the panelists who presented today. It's time for our discussion. So if all the panelists could go ahead and turn on their cameras, and we can go through the set of questions. Okay, so the first one is a question for the panel. What can we do in the future to have an oversight, particularly the welfare of mice and rats used in testing for test facilities that do not have any PHS funding and AAALAC accreditation? Nicolette, did you want to answer this first?
Yeah, I did. And I one of my favorite things to say is there are better reasons to do things than just because feds require you to do it. One of the things that I think is really important is this is if you apply the same standards to all of your mice and rats, what you're doing is you're sending a message that the welfare of all of the animals matter at your institution. It's a great talking point for the benefits of your research and how animals are protected. So I think it's a great public relations point. And for the AWIC especially, one carrot you can give your investigators is that, you know, doing this, if everyone follows the same SOPs, and they're standardized across the board for all mice and rats, regardless of funding source, that's more efficient, less likely for people to get in trouble and have a non-compliance or affect animals that may be PHS funded. And that requires a letter to OLAW. So, you know, there are lots of ways to mitigate risk by doing it this way.
Yeah, great. Gwen, do you have anything to add?
I do not, no.
Gary or Susie?
Yeah, so go to our website, look up how to become accredited. And if you have any questions, call me at 301-696-9626.
Thanks, Gary. And assuming Susie doesn't have anything to add.
No.
Great. Next is, does AAALAC require its accredited facilities in non-US countries to provide EE environmental enrichment / well resource housing for rodents and other animals, or are there countries where this is not required for accreditation?
In a word, yes. So the standards, references, position statements, those apply to all accredited programs. You go look at our FAQs, we have information about environmental enrichment. The guide speaks to that. So those expectations would apply to any accredited program.
Great. Next is kind of a partial question. Non-accredited institutions are eligible to apply?
You know, I think that question popped up when I was talking about the Global 3Rs Award.
I think
And so the wording on the website says any researcher, then there's some footnotes and some disclaimers. So look at those details. There's no link to being an accredited program and applying for the Global 3Rs. And that's really the answer I think the person was looking.
Okay, question for Susie. They missed the contact information for the Food Safety Consortium. Can that be reiterated?
What is it again?
The person missed the contact information for the Food Safety Consortium. Could that be reiterated? And you can give that to us.
Okay, all right. I mean, yeah, I'm not exactly sure what you're talking about. Are you talking about our public-private partnership or what we're doing for Ilmerac, probably, yeah. Okay, okay, I can get that for you.
Okay, great. One for OLAW. What would you recommend IACUCs do in regards to the Arrive Essential 10?
So I guess the first thing would be just publicize that guide notice and send it out to your PIs. You know, if they see that it's important to NIH, I think that is powerful because so many people have or want NIH funding. The other thing I would say is another little carrot that you can dangle for the PIs is that, you know, if you use the Arrive Essential 10 to describe your protocol, if it's built into the experimental design, that means the IACUC is likely to understand the protocol a lot better when it comes to approval, and it's going to be less likely that they're going to get a lot of questions.
Yeah.
So that's something to consider.
Yep. So there's a lot of pressure on IACUCs to encourage and make sure PIs are adequately considering the 3 Rs. But how do they overcome some of those barriers with this? For example, you know, not being knowledgeable about every alternative or NAM, questioning senior PIs, protocols, I guess, i.e. office politics. Do you all have any recommendations for that?
I'll just jump in and say that it's a tough process, and we can talk a little more about certain approaches that you can use at the final session of the day, because we're going to be talking about how to adopt certain R's. I think it's going to be incremental progress, and it's going to be just reiterating how, as more knowledge accumulates about refinements and replacements, and we have more emphasis on reductions, I think tying it back to how this will benefit their science is really important. And as we've learned from the past 2 days, there are really approaches, as Dr. Nimi mentioned, these are not alternatives. These are actually excellent approaches that improve the science in their own right and are not just something to use instead of animals. They are powerful approaches. So, you know, trying to personalize it and relate it to their studies, and also being receptive to the concerns that, well, yes, it's validated, but it's not validated for my work. How do I know it's going to be okay for my work? These folks rely on their publications, and they rely on their past track record and past studies. So one of the things that was mentioned on day one were these helpathons. Hopefully we'll be seeing more of those in the future, but, you know, being receptive to the idea that just because an alternative exists, it may not be appropriate for that particular study, or if it is, more work needs to be done to really see how that will fit into the workflow. I think that's all important.
Yeah, and I think you mentioned something at the end that's very powerful, is recognizing the limitations of these NAMs and alternatives and being realistic. Gwen or Gary or Susie, do you have anything to add?
I can just add that, you know, one of the tenets, if you will, of AAALAC is continuous improvement. And in that realm, you know, a mindset could be, don't ask what I have to do, but think about what I can do.
Susie?
I'll add that any alternatives that are validated, generally there's something put on a website, either on ICVAM or on one of the agency's websites, as to what's validated and what the protocols are. So,
Yeah. And Gwen?
I think that, again, finding ways to educate IACUCs about the alternatives is really key. A lot of IACUC members do the IACUC, their IACUC responsibilities in addition to a full-time, sometimes full-time plus job. And so there is a time stress in terms of being able to learn about the new alternatives for a lot of the people who do this work. I also think that I've seen a challenge with PIs subcontracting out their alternative search and facilities providing resources for someone else to do the search and then just plugging it into the protocol. Because that, to me, I feel like undermines the whole purpose of the alternative search. That's another problem that I've noticed out there. It's not non-compliant, but it's but I feel like it does kind of undermine the whole point.
Yeah, that's a really good point. Thanks. I think this one is also for you, Gwen. Do you think the definition of animal will ever change under the Animal Welfare Act, and mice, rats, and birds bred for research will be covered?
Well, we have seen it change. It was just a semicolon, I believe, that was changed, and now we cover birds, and our bird standards have been published. But it's not, it does exclude birds that are purpose bred for research. I'm not aware of any discussions right now that are looking at including mice and rats and birds bred for research. But it is something that seems to come up from a lot of different groups at different times. I'm sure we'll be revisiting it. I don't know if we'll be regulating it.
Yeah, it always seems to come up in these types of conversations. So we never know what the future will hold. Another one for you, is there a place on the APHIS website where the pain categories are explained or defined? I feel like this is a really good question. AWIC gets a lot of people asking us this question.
On our website, we have we have a page for the research facilities to submit their annual reports. And on that page, there are resources. And the resources do go into more detail delineating the different pain categories and what falls, and with examples of what would fall into one category versus another.
Okay. Another one for you. Can you please expand on the animal reuse for procedures that are not major operative procedures? Does USDA look at animal reuse justifications of institutions for non-major operative procedures?
We don't have any regulations that specifically speak to it. Our regulation, we just have the regulation on the major, multiple major operative procedures. However, there are studies that don't involve multiple major operative procedures that do have a significant impact on animal health and welfare. And so while there's no regulatory, we wouldn't look at that at this point in time because there are no regulations that speak to that. There's still an animal welfare concern depending on the intensity of the protocols that they're on before they're reused and what they're reused for.
Yeah. Okay, so what do you think needs to be done to push institutions to use animal alternatives when in vitro alternatives are not explicitly stated in the CFR as approved methods by the FDA? For example, MAT or MAT testing to replace in-life pyrogen testing in rabbits. Maybe a question for Susie or Gwen.
I was gonna say, was that for me or was that for Susie? I think that was for Susie because it's asking about the FDA.
Yeah. Susie, do you have any comments? I can also reintroduce the question.
Can you repeat that again?
Sure. What do you think needs to be done to push institutions to use animal alternatives when in vitro alternatives are not explicitly stated in the CFR as approved methods by the FDA? For example, MAT testing to replace in-life pyrogen testing in rabbits.
I'm not sure that they put, I'm not sure any animal tests are in the CFR either. I mean, we have guidance on them and stuff. I think we can encourage people to come in and talk to us, but we can't make them do it. I mean, I think in some sense, people don't use them because they don't really know they don't want to raise any concerns in an application. So we have put a lot of work into in vitro microphysiological systems, including working with IQ Consortium, MP affiliate, for several years, but we have yet to see anyone putting any data in an IND. So I don't know other than to say we would accept them. And I don't think we're going to, if you put something in a regulation, it's very hard to change. So you put it into guidance, which is your best thing. Put in a regulation, it takes years to get in. And then if it's out of date, it's going to take years to get it out. So that's not really a good idea. So.
Okay. Thanks, Susie. We have some additional questions, and I think this will apply for everyone on the panel. We know minimization of animal numbers and minimization of pain and distress are required for both USDA and OLAW, but how does provisions of positive experiences factor into the regulatory requirements? Nicolette, do you want to go first?
Yeah, so I talked a little bit about this during my presentation, that the US government principles and the guide enrichment requirements discuss things like behavioral abnormalities, self-injurious behaviors as something to be avoided. And that can be done through achieving complex environments that provide species typical opportunities for species typical behaviors. These are all in addition to preventing the negative consequences like SIBs. They also provide positive behavioral experiences. And I think that it also, my personal opinion is that it also prevents compassion fatigue in your staff. So I think that we do address it somewhat indirectly, but when you are addressing things like social housing for social species, complex environments that allow species typical behaviors, and you're managing all the other things that go into good welfare, like a good veterinary care, appropriate diet, and all of that, you are maximizing the chances for those animals to have a positive behavioral experience. I think some of the things, I think earlier in the day, we talked about Kathy Shipley's work, training your staff to handle these animals in a welfare appropriate way, using positive reinforcement conditioning wherever possible instead of restraint for handling procedures. I know it can be a big ask to get everyone trained and on board, but if you look at it also, if you look at compassion fatigue as part of the equation, I think the benefits are even more than people realize.
Yeah, agreed. Gwen?
The standards for non-human primates include the environment enhancement to promote psychological well-being. The regulations for dogs include the requirement for exercise, and in that requirement, it does support social housing by if the dogs are socially housed and they're pair housed, it allows facilities to house them more efficiently and allows them to get the exercise. So that those are two areas that where the animal welfare regulations support doing things that are positives. I think when we look at the positives, a lot of times it delves more into the behavioral side of things and behavioral needs than the physical needs. And I think that a lot of the stuff in the Animal Welfare Act is primarily based on meeting those physical needs. We're getting more and more into the behavioral side. And again, I mentioned that we had a notice of public rulemaking go out that we are looking at expanding environment enhancement for all covered species. So that would also, again, contribute to that positive. But one thing that's really a challenge is trying to regulate compassion is as difficult as it is to regulate people considering an alternative. It's hard to measure whether that line is being met or not from a regulatory legal standpoint.
Yeah, that's a really good point. Gary, do you have anything to add?
So a phrase comes to mind. There are two types of people in the world, people that put things into categories and people that don't.
Yeah.
So as I look at the purest definitions in Russell's and Birch book, they talk about direct inhumanity and contingent inhumanity. So I was talking in my talk about direct, the 3 R's. The contingent inhumanity, I think, is what you're referring to here, talking about good veterinary care and animal welfare and things like that. So in that regard, there are references, resources on our website in the same categories like I talked about earlier. FAQs, the guide has a lot of information about this. So we certainly emphasize and promote those types of, support for the animals in that space. I don't know if that answers the question or not.
Yes, no, it does. And Susie, do you have anything to add before we move on?
No, I don't.
Okay, next question's for OLAW. So if a grant has been approved, but the AWIC has concerns about the ability of the experimental design to meet the research goals, or if the AWIC is aware of a refinement, reduction, or replacement method that are likely to be appropriate for the research, can this be brought up with the PI even though the grant has successfully undergone peer review?
I'll give the short answer first, and I'll give the long answer. So yes, it is within the AWIC's authority to require changes to a protocol before grants awarded. And AWICs can certainly come to OLAW because we can assist with questions. But in addition, if there are big changes being requested, contacting the grants management official through appropriate institutional channels can be really helpful, because some of these changes may involve a change in scope. So it's always good to check with them if you're asking for some larger scale changes that could be categorized as change in scope. That being said, I realize that this is a really difficult thing to do. And there's an art to it, I think, and maybe asking questions is a good way to open up the conversation and then hear how the PI responds and go from there. Because we did discuss that there are certain R's that are generally widespread and mentioned for a lot of different applications, but the real key is figuring out how it will work for that particular PI's application. So there is an art to this as well as a science.
Yeah, great answer. Okay, next question is for Susie. Does the FDA require animal testing for drugs in development before allowing clinical trials? If not, what are the best practices and FDA reference resources for submitting data from non-animal models or methods?
Well, FDA doesn't require, like I said, there's no requirements in the regulation for animals in preclinical, but you have to, generally you have to use some studies to demonstrate you have confidence to put it in a first in human. They work a lot with ICH, and if they follow ICH, that's where they're discussing what could be part of a preclinical application. So that's ICH is like global, and they have part of it for and those again are guidelines. So there's no actual requirements, but you know you have to there's things you have to demonstrate.
Okay. Another one for you, Susie. So while the FDA Modernization Act 2.0 encourages the submission of non-animal research data, do you see it making any drastic changes or, quote, moving the needle to utilizing non-animal models? Are there any educational outreach or guidance planned regarding the act?
Well, as I said before, I don't think it's changed. We already were emphasizing on an agency basis, using non-animal models. We have a FDA alternative website where people can come in. On that website, and we have an alternative animal work group. There we talk about it. We invite people to come in and bring new methods to us. We probably had about 70 different people coming in and presenting new methods, as long as they meet certain criteria, mature methods that might have a context of use. So at least once every couple of weeks, we work on, I chaired the last MPS, co-chaired the last MPS summit. We have another one coming up. So I think we do a lot to encourage people, but as I said, people can, it's the same with acute tox testing. We don't require it, but people do still put it in,
Yeah. Okay. And one more question for you, at least for a while. Does the FDA have metrics to show how many applications you're getting that use alternatives or NAMs?
No.
And if not, are any planned? And if so, what kind of metrics are being considered?
The metrics that we have been asked about metrics, including counting animals, we don't believe counting animals is a good metric. Similarly, the data that we get is not our data and it's confidential. Most of the INDs come in, if they're not approved, we can't even say they've come in. So it's not our data. We're not going to count animals. We think what we're trying to do is to count accomplishments. And so on our website, we put papers, we put talks, we put a lot of different things that we've tried to emphasize accomplishments. I know that we're asked a lot about counting animals, but you can say we didn't require this or this or this. We didn't require a repro study, so we saved 20,000 out, but they could have done it anyway, and they just didn't give it to us. So I think we have a lot of different programs going on, and like I said, you can track everything on our public website on alternatives at FDA. I mean, CEDAR just put out 2 papers on their gaps where they like alternatives. We're working on a paper. Other centers are encouraging to do that. I talked at ICFAM today. CTP talked at ICFAM today about what we were doing. So I think we've talked a lot about it and demonstrated what we want to do.
Okay. Yeah, it's a good point about you're not aware of the data that you're not given. So if animals were saved, but that's not provided, yeah, it's hard to know. Okay, one for Gwen. What tools and information do you provide to the boots on the ground inspectors to ensure that institutions and facilities are adequately considering the use of alternatives?
Well, it is included in our training for our new inspectors when we are training them on inspecting research facilities. And then the information that they're trained on is summarized in the inspection guide. And that is kind of the backbone of what they are looking for and paying special attention to.
Okay. There's a question that came in the Q&A. Isn't it true that many preclinical studies use NAMs happen before the data used for FDA submission is collected or submitted? Susie, I think that's for you. I can.
Okay, I think you're talking about preclinical screening of different drug components or different chemicals to get the optimal one to put forward. Data that's not submitted to FDA. We suspect that's where they're doing some chip work, but that's not given to FDA, so we really don't know what's going on. We kind of know, but again, it's not required to submit it.
Thanks, Susie. Similarly, Gwen, how well do you think the inspectors are scrutinizing or reviewing the alternatives literature search portion of the animal use protocol? What kind of feedback, if any, do you provide or does the inspectors provide to help improve the searches?
I think it's there is, being human beings, there are inspectors that are really, really good at it, and there are probably some out there that are not as strong in that area. You get that anytime you work with people, a large number of people, there's going to be some variability. I just, like in the last month, was on an appeal team looking at a citation that we gave for consideration of alternatives. So I know we're citing it because I was just reviewing a citation on it. We do, to some degree, leave the adequacy of that search up to the AWIC, and as long as it's within the professionally accepted standards, we're finding them compliant. And so it would have to be very clearly not meeting that requirement.
Okay, so you're saying that level of detail really falls under the scope of the AWIC review?
Just to some degree, yeah. Because when we're looking at it, this is something that's already been reviewed by the AWIC and approved by the AWIC. So we're looking at something that's already been approved by their internal review program. So if it's something we're gonna find non-compliant, it's usually something that is fairly black and white, not meeting that professional standard, not meeting the requirement to provide information about that search.
Yeah. Okay, thanks. Okay, one for AAALAC and OLAW. So in your presentation, you mentioned the use of lower phylogenetic species being considered as an alternative. Do you feel that a lower species truly represents an alternative, even with new research coming out potentially saying otherwise? Nicolette, do you want to go first?
Yeah, this is a really good question. That language is in the guide on page 5. And I think this is a statement that needs to be interpreted in light of the most current scientific information. And I really do think that we are entering the golden age for cognitive and behavioral studies. And I'm a little bit of a behavior geek. So one of my favorite papers was a 2011 paper by Peggy Mason and her group. And they showed that rats will forego a food reward in order to rescue a restrained conspecific. And, I was thinking, yeah, but everybody knows rats are nice. Anybody who's ever worked with them knows they're wonderful. But there was a 2019 paper by another group that showed the same thing in rats. It was a little different. They ate the food reward first, and then they rescued the conspecific. But I think this type of thing would have been unheard of 20 or 30 years ago. And what we're seeing now is that all of these complex behaviors that we associate with higher species, it's actually falling on a spectrum. And there's more and more information showing that many, many more animals are capable of performing complex, higher level functions than we originally thought. It used to be that theory of mind was only for great apes and elephants, maybe, and now we're seeing them in a host of other species. Used to be that only chimpanzees were using tools aside from us, and now we see a multitude of species that use tools. And I think that as we go forward, we need to go where the data takes us. I also personally have a little bit of an issue. This is me speaking from my personal opinion with phylogenetically higher or lower, because from an evolutionary perspective, that doesn't make a whole lot of sense. Are you talking about the most recent common ancestor, or are you actually talking about their fitness? In either case, it doesn't really apply. But I think in the past, what they were trying to do were to take animals that, and I can only guess, that showed more evidence of sentience. And now we're seeing that other species that previously were not really evaluated for sentience are showing much more capabilities in that regard. And I think we need to take that into consideration.
Yeah, and I think your one example you might be alluding to is, zebrafish, which we now know. We had a speaker at a symposium a few years back who demonstrated over, multiple research studies that they do have preferences. They, do have choice or have choices when given them and feel pain. So I think we've come a long way in terms of that. Gary, do you have anything to add?
Just two thoughts, and I'll start with reading our definition of animal from the rules of accreditation. So our definition says all animals used or to be used in research, teaching, or testing at accreditable units are to be included and evaluated in accordance with the standards set forth in section 2F of these rules. This includes traditional laboratory animals, farm animals, wildlife, and aquatic animals. Non-traditional animals, inclusive of invertebrate species, are also included where they are relevant to the unit's mission. So from our perspective, we're looking at the range of animals. And then the final comment, the second thought is that, if we look at what Russell and Birch penned in 1959, they talked about full replacement with replacement, but we've kind of morphed, I think, as a society or as a group into partial replacement. And that's where I would talk about lower species. Thanks.
Okay, great. Thank you both. One more question for you, Susie, and you touched on this in your presentation, but just to see if you have anything to add. How does the FDA work with PIs on submitting research data? Is it through the research process or really just towards the end when they're getting close to submitting their data? Are there any new initiatives or planned initiatives to improve collaboration?
Are you talking about research data as part of an application for
Yes.
we encourage people before they put any application in any of our programs to come in and talk to the FDA about what they want to submit. They have several different pre-IND and pre-phase 1 and phase 2 testing meetings that they and then other centers have that too, to come in and talk to FDA, make up an appointment and talk to us. But it's not a requirement, but we are encouraged for research projects at academics and others. We do oftentimes do some type of research agreement. People mentioned microphysiological systems. We have a CRADA to move those systems in several of our labs, and we use those as not only doing research, but learning tools for our regulators, so that when they see these types of methods in an application, they understand them better.
Okay, thanks, Susie. And I don't see any other questions in the Q&A. So with that, I'll go ahead and wrap up. I want to thank our panelists again, not only for giving a great presentation, but also sticking around to field all of these questions over the last 30 minutes. We really appreciate your time. And thanks to the attendees for sticking on. We'll take a 15-minute break. If you could report back at 3:15 p.m. or 3:10 p.m. Eastern time. And then we'll close out with our last session with Dr. Liz Nunemaker, who's going to discuss some of those 3R scenarios that you submitted over the lunch hour.
Thank you.
Yeah, thank you, Gary. Thank you, everyone. Let me know, Nicolette, if I missed anything and what I should be announcing to the audience.
Sounds good, Jesse. I think that's good. We'll have some last minute announcements at the very end too.
Yeah. Okay. See everyone back at 3:10 p.m. Thanks.